Hello everyone, welcome to Lovebirds Genetics. In today's video, we are going to learn how to use Lovebirds genetic calculator. This video is going to be about Fisheri lovebirds, that is Agapornis Fisheri, and in coming next videos we will be learning more about how to use genetic calculators for Agapornis rosicolis or Agapornis personata. Make sure you follow our channel, subscribe, like and comment in order to get more such interesting videos on your feed. Let's get started. So without wasting time, let's start today's video. So first of all, you are supposed to go to your Chrome browser or Safari browser, and you need to type genetic calculator for lovebirds. As you can see, the very first one is white eye ring group lovebirds. You need to click that. Now you will see an interface like this. This is the whole genetic calculator which you are going to use for making n number of varieties which you want to produce. So as you can see, let's go through each column one by one. So as you can see, here are some symbols. Here are some symbols. V stand for violet, D for dark, BL for blue, and like that. OP for opaline, PI for pied, and all that. Then we have MT for misty, ED for edged, slaty, SLT, PF pale fellow, and Ewing. Then we have here mutation names. So we have violet, dark blue, turquoise, that is par blue, opaline, dome pied, recessive pied, NSL eno, which means non-sex linked eno, in which we have deck, which is dark eyed, cleared, pastel, bronze fellow, dilute, misty, dominant edged, Ewing, and so on. And we also here we have inheritance mode. So basically inheritance mode is that how that particular mutation is getting inherited from parents to the offspring, whether the mutation is on sex chromosomes or whether the mutation is on autosomes. So that is inheritance mode. We have here when it says in it is referring to incomplete dominance. Then we have RE for recessive, CD for codominance, SE for sex linked, DO for dominant, RE for recessive, CD codominance, and again all this. So all the abbreviations are mentioned here. As you can see, RE for autosomal recessive, SE for sex linked recessive, DO for autosomal dominant, N for autosomal incomplete dominant, then we have CD, which is codominant, and L for multiple alleles. Now there is something else. Okay, so here 1.0 stands for the male bird. As you can see here, 1.0 stands for the male bird and 0.1 stands for the female bird. Always remember that. So I mean, when you are putting up the details of your bird parents in order to get to know the baby's mutation or how are they going to look like, so you need to put your parents details here so that we get to know what the babies are gonna be. Again, so there is something called visuals and there is something called splits too. So when I said visuals, it means that how your bird looks from externally. I mean, what is the morphology of your bird. And splits too it means that how if your bird is carrying genes or alleles for any other mutation inside their DNA, so which you can't see actually, but yeah. So I think visuals and splits too is clear. Visual is what you see from outside, and splits too is kind of carrier which means that if your bird is carrying any other allele or any gene or for any other mutation. So let's dive into this thing. Now, as you can see, there are two more things, which is called visuals only, and in bracket we have no splits. Then we have show genetic code. We actually don't have need this show genetic code, but yeah, it is kind of more genetics, but it's okay if you don't have to learn about this stuff, but you should know about the visuals only or no split. So it means that when you type the information of parents here in this table, then you are going to see only the visual, that how the babies are going to look like externally. It will not show the splits. Let's do it. And one more thing which you need to know is that if you don't fill up any one parent's detail here in this blocks, either the male 1.0 or either the female, then it will it's a default setting that it's gonna assume that you are talking about a wild green bird. In fisheries it is just your green fishery, and if it is like your rosy peach faced lovebird, then it's gonna assume it's a simple rosy green beach faced bird. That's it. So that's the default setting. So you have to put your details of the parents very accurately here in order to get the best results. So okay, let's start doing small tutorial videos. I mean small, let's take some examples and do it now. Let's do a simple cross and see how to use this genetic calculator. For our first cross, we are not putting anything on this parents column. So let's say if we are not putting anything, and we just hit this button which is generate, then it shows, as you can see here, we have results genetic calculator 1.3 white eye ring group. Then this is this is 1.0 green crossed by 0.1 green female, and then we have here percent from all 1.0 and percent from all 0.1. When we say percent from all 1.0, it is talking about the male babies which you are going to get, and when we say percent from all 0.1, it is talking about the female babies which you are going to get. So as you can see, we have put nothing on the parents column, parents rows, or any details of the parents here, but still we see green and green. This is what I meant when I said that if you don't put the correct details, or if you forget to fill up the details for any one parent, then the genetic calculator is going to assume that you are talking about the green bird. So in this case, when you are breeding a green fisher with a green fisher, you are going to get 100% green fisher babies. You are not going to get any other baby. Let's go back and again let's do another cross. This time let's assume, okay, so this time we are doing another simple crossing. Let's assume we have a green fisher male and we have a Lutino female. So for Lutino, you are supposed to select this thing which is NSL Eno, and you just click it. You have put the details for the female, and we are not selecting anything for the male because our male is just a green fisher. Then you generate. Now you see again the male for our this crossing was a green fisher, and the female for this crossing was a Lutino fisher. So here we have percentage from all 1.0, percentage from all 0.1. These are male babies, these are female babies. As you can see, still you will be getting all the green babies, but you see NSL Eno here, right? NSL Eno, it means that all these green babies are going to carry the alleles or the gene for Lutino fisher. It means that they are carrier for Lutino mutation, right? Let's go back. Again, let's do one more calculation. Let me reset it. Do a crossing involving blue parents. Let's assume you have a blue fisher and you have a blue female. Then you are going to get again all the blue babies, blue 100% blue males and 100% blue females. Let's go back again. Let me reset it. Let's assume we are working again with a blue bird here. Let's consider we want to select blue opaline male and just a green opaline female. So when you want to fill up green opaline, I told you initially that you don't have to select anything for a green bird, but you know green opaline is having, apart from being green, it also has a mutation called which is opaline. Okay, so you need to select the opaline. So for the female we have green opaline and for the male we have blue opaline, and we generate the results. Now you see our male was blue opaline and our female was green opaline. Male babies, female babies, and as you can see all the male babies are going to be green opaline, but they will be carrying the gene for blue mutation. And again all the females are going to be green opaline, but they are going to be carrier for blue mutation. Now let's do one more thing. Let's do cobalt opaline. Okay, as you can see this row which is dark, as when we say single factor we are actually talking about this which is cobalt. When we say double factor we are actually talking about mauve or this black bodied fishery. So how to get back, we need to go back. Okay, so here let me reset it. Yep, now let's do a simple pairing which involves cobalt. Okay, as I mentioned, SF stands for single factor. In blue series, single factor blue is cobalt, and double factor blue is your mauve or mauve. We have one, and this is our parent which is cobalt, and again, and here we have blue opaline female. So yeah, let's generate. Now this is interesting. So you had your cobalt blue male and you had blue opaline female. From this you are going to get these results: male babies, female babies. You'll be getting 50% normal blue fisheries which will be carriers for opaline, and then you'll be getting some cobalt fishers which will be carrier for opaline. Then you'll be getting these birds like the female babies which will be simple blue fisheries and simple cobalt fishers. Let's do our last cross, and we will be good with this today for today's video. So let me reset it. Sorry, we didn't. Yeah, reset. Yeah, now let us do let us see how to select, or now let us do a crossing for Lutino with an albino. So here on genetic calculator, you know that NSL eno is like non-sex linked eno, and it is kind non-sex linked means the mutation is on autosomes, not on sex chromosomes. And eno is a gene which is responsible for reduction of eumelanin, which actually converts a green bird into a yellow bird and a blue bird into an albino. So this is kind of basic. So for we know that albino is a blue bird, so when you are selecting the albino bird, you are supposed to select blue as well, and apart from that you need to select NSL eno. And for this crossing we are taking Lutino Fisher as the male and the albino female. So let's generate again. This is interesting. So when you have a Lutino Fisher male and you have a albino female, the results are this. You will be getting all male and female babies which are Lutino, visually which are Lutino, but they will be carrying the blue genes. So if we continue with these births, let's assume, let me reset it. Yeah, so we saw that we got Lutino Fishers male which were split. Here we have splits too, which were splits for blue, right? Again we had here female babies which were Lutino Fishers visually, but they were split for blue. And when we breed these babies together, we get this thing you see here. So we had Lutino Fisher male split for blue and Lutino Fisher female split for blue, and male babies, female babies. As you can see, you will be getting 25% Lutino Fishers here, which is NSL eno. Then you will be getting 50% babies which will be Lutino Fisher visually, but they'll be carrying the genes for blue. And then you'll be getting 25% which will be your albinos. Then for the female babies as well, the same thing follows. You'll be getting 25% NSL eno, 50% NSL eno, which is like Lutino Fisher split for blue. Then you'll be getting albinos. So this is how genetic calculator works. We will be doing more calculations in our next video. Let me know if you have any specific crossing in your mind which you want me to work on and make a video. I will be more than happy to do that for you. And please do comment which type of births you guys have, and are you working on any specific mutations, and if you want me to make any specific videos on any mutation, how to produce that, and if you need any help, I'm always available on Instagram, Facebook, so you are just a message away from me. So guys, I hope you liked this video today, and please do subscribe, comment and like the video. Let's meet in the next video.